Researchers at North Carolina State University have developed a portable technology that allows farmers to identify plant diseases in the field itself. The handheld device, which is plugged into a smartphone, works by sampling the airborne volatile organic compounds (VOCs) that plants release through their leaves. All plants release VOCs as they breathe, but the type and concentration of those VOCs changes when a plant is diseased. Each disease has its own signature profile of VOCs. So, by measuring the type and concentration of VOCs being released by the plant, we can determine whether a plant is diseased and if it is diseased, which disease it has. Current disease identification techniques rely on molecular assays, which take hours to perform and most importantly have to be done in a lab. Getting a sample to the lab where the sample may have to wait to be tested and can delay disease identification by days or weeks. This new technology will help farmers identify diseases more quickly so they can limit the spread of the disease and related crop damage. Here is how the technology works. If a farmer suspects that a plant may be diseased, he can take a leaf from the relevant plant and place it in a test tube. The test tube is then kept for at least 15 minutes to allow the relevant VOCs to accumulate. After this incubation period, the cap is removed and the farmer uses a narrow plastic tube to pump the VOC laden air into a reader device connected to a smartphone. The air is pumped into a chamber in the reader that contains a paper strip. The paper is embedded with an array of chemical reagents that change color when they come into contact with a specific chemical group. By evaluating the resulting color pattern on the strip, users can determine the nature of any plant disease that may be affecting the plant. In proof of concept testing, the researchers demonstrated the device's ability to detect and classify 10 plant VOCs down to the parts per million level. They were able to detect the late blight pathogen that caused the iris famine 2 days after tomato plants were inoculated with the pathogen. Researchers could also distinguish tomato late blight from two other important fungal pathogens that produce similar symptoms on tomato leaves.